All right, I think we're all set. So welcome everyone. This is the very last session of this edition of the Biocatalyst Innovation Lab. So this is the final pitching event, the moment of truth. And today we will have teams present their work, the ideas and the projects that they've been working on for the past months. So after a short introduction, we'll have the team presentations, after which the jury will convene in a breakout room to decide on the winners. Meanwhile, we have a couple of announcements for the Biocatalyst participants, which you might find interesting. And finally, we will have the awards and the closing words. So in total, it should take about 2 hours, 2.5 hours, depending on how smoothly we move through the presentations. So we've always envisaged the Biocatalyst Innovation Lab as a sort of hybrid with both educational and competitive elements. That's why we've had both guest lectures and self-study and background reading, coupled with teamwork and problem solving. And the latter part we still call a hackathon, even though it has grown to be much bigger than a typical hackathon that you would encounter. And the 5,000 euro prize for the latter has been provided in cooperation with the Riga Technical University Students' Innovation Grants. So for the purposes of this program, we view biotechnology in the broadest possible sense. So anything that can be solved by involving living organisms. And for the challenges that were given to participants during the Innovation Lab, we divided the topics into 4 broad tracks: health and well-being, food materials, circularity, and ecological problems, as well as improving the toolkit of biotech itself. We had approximately 100 applicants from 7 countries and more than 30 universities, and of course also people who are already past their studies, of which we formed 15 awesome teams. And so the 10 teams who made it to the finals, I congratulate you, you're here, and now is the time to show us what you've got. During the last 6 months, we've been quite busy. So we had both guest lectures by interesting speakers. We had background reading quizzes. We had team building sessions. We learned to use, we practiced to use design thinking in identifying needs and looking for solutions. We applied the creativity. And so now is the final session where we present everything that we've learned or everything that we've created to the jury. And it is my great pleasure to welcome the jury members. I will start with Anna Ramatastunda, whom you already have met during our last guest lecture. Anna is the co-founder and CEO of Alternative Plants, a biotech startup from Riga, and she's also on the board of Malaria Cosmetics, which is one of the largest cosmetics producer in the Baltic states and Eastern Europe. And next, we have Professor Talis Juhna, who is the Vice Rector for Research at Riga Technical University, and also the head of the Joint Biotechnology Study Program at R2 and Latvian University. And next, we have Rolando Delgado, whom you have met during the kickoff session. Rolando is the head of business development for MGI, the gene tech multinational giant. Next, it is my great pleasure to have Nadav Rosenberg with us. Nadav is a venture investor and also partner in the Entrepreneur First program, which is one of the largest talent development programs in the world, which helps people achieve their dreams by founding new companies. Next, we have Anna Janberger, who is the leading expert technology scout at the Latvian State Investment and Development Agency. who have been very helpful to us during the organization of the Innovation Lab. We have Dr. Petris Trapenseris, who is the head of Department of Organic Chemistry at the Latvian Institute of Organic Synthesis. And last but definitely not least, we have Andrei Nahkala, who is the head of corporate affairs for Finland and Baltics for Biogen, which is one of the top biopharmaceutical companies in the world. So right now, I would like to ask the jury members, just in case, if you all have the access to the jury file. If not, please let us know and we will send you the link again. Please also make sure that you are choosing the marks in the tab with your name on it to avoid any confusion. So if you have any questions in the meantime, you can always ask us in the chat or contact us personally, or just speak up. All right, without further ado, here is the setup for today's session. You have already seen these before, but let's quickly run through this again. So we will have teams presenting their solutions. Each team will have 5 minutes per pitch. We will tell you when the 4-minute mark arrives, and we will stop you at 5 minutes, and you will have the chance to finish your sentence. So please don't click through the rest of the slides if you are stopped. Just try to gracefully kind of wrap this up. Each team will also have 2 minutes for questions from the jury. So since it's not a lot of time, please consider it as 1 or 2 questions maximum. Otherwise, we can be way over time. You have all seen the evaluation criteria by now multiple times. The jury members have the criteria under their evaluation tables in the file. And the sequence of the teams will be according to their team numbers during the program. So you all know your team numbers, and I will also be announcing each next team. One person from the team will will share their screen and present the pitch. The other team members can also, of course, take questions. And as a reminder, again, please be sure to turn on your cams when you're presenting. Both the person presenting and the team members, we would like to see your faces. All right. Are there any questions so far? Great. Let's move to the pitches then. And the first team to present will be Desi for Medi. I've seen you here, guys, so please share your screen. And the floor is yours.
Good afternoon, everyone. We are here to present you our first aid kit and app for vector-borne diseases, and we are Laima, Teresa, and Maria. Vector-borne diseases are human illnesses caused by parasites, viruses, and bacteria that are transmitted by vectors. Examples of vector-borne diseases include dengue fever, West Nile virus, Lyme disease, and malaria. Agenda 230, the third goal of it, sustainable goal, is good health and well-being for everyone. And in order to achieve that, our common goal is to end epidemics of vector-borne diseases. And the reason why we want to do so is because the risk is 80% of world's population is at risk of one or more vector-borne diseases. The burden is around 17%. Mortality is very high, about 700,000 per year. Case study, malaria. We have 229 million people dying from malaria annually. They are affected by malaria annually, and 409,000 of them die. 66 67% of these people who die are children under 5, and around $3 billion US dollar was spent on malaria in 2019. The most problematic stuff in Europe are ticks, and there are around 200,000 to 400,000 people affected by that. And if you diagnose it early, you can treat it with antibiotic, but if it's too late, then it can be problematic. And here you can see that vector-borne diseases, they're on the rise from 2004 to 2016. It was a huge rise, and the most mortality is in Africa. So our solution is an app combined with biosensors. I want to catch these insects in the air. So we want to use remote sensing. And for quick solution, we propose 3D printing. And we want to use AI to analyze the information from the sensors. The biosensors market is predicted to be 36.7 billion by 2026. And around 15% of that would be for vector-borne diseases. So we want to cover the whole globe, and for Europe, we are most interested in ticks, dengue, West Nile viruses, and then we couple them to sensor and analyze the results. And we also want to offer a first aid kit. It's either all included or pick and design yourself. Deployable platforms are wearable, iPhone, Android, laptops, iPads, and the kit is an app in the form of a USB. Work in progress is vaccine and test kit with focus on epigenetics, indoor residual spray to kill and get rid of those insects, filter to make sure that we have clean water, hygiene control for food, tweezers and skin disinfectant. And the vaccine and test kit will be based on epigenetics. Epigenetics is controlling how genes are switched on and off. And right now we do have a vaccine that is being tested successfully, 77% efficient for malaria, but people would like to investigate why it's not 100%. And our hypothesis is that it might go through epigenetics. And we know that methylation states and response is one of the reasons why hepatitis B vaccine is not 100% efficient. We will do first computational studies and simulate our ideas before we test it in the lab. And in order to speed up the stuff, we will use supercomputers. And our business model, well, we try to have a future IPs on the innovation ideas that we have, and human resources, and activities are problem solving and preclinical drug development. Thank you for your attention, and a big thank you to the arrangers of the hackathon.
Thank you, Maria, and thank you, Team One. Do we have any questions?
One question from me, Peter Strapen series. You mentioned about epigenetics. What others do you mean when speaking on methylation issues?
Well, we had to sequence the genomes of patients, and probably they have different methylation, and that is why some vaccines are going to be efficient for if they have we have to look at immune system methylation, stem cells methylation. We have to look at different cell lines and see how that is done. And for this hepatitis B vaccine, so they could see like an immune system if they had different methylations when they sequenced the genome of the patients, so they got different responses. And methylation is like it will like change how active or inactive your immune system is. Because the vaccine idea is that you activate your own immune system. And one of the reasons why vaccine maybe not be efficient is that immune system. Either is not activated enough or it is overactivated. And we want to see if it's due to individual methylations.
What level do you mean? Histone level or DNA level?
Histone levels.
Okay, thank you.
Thank you, Patrice. Any more questions to team one? All right, then. Thank you again. Thank you, Maria and team one, Desi for Mehdi. We will now move to the next team, team number two, which is called VACO.
Good evening, ladies and gentlemen. This is Hiba from Team VACO. I'll be sharing my screen in a bit and let me know if it's visible clearly. Is it visible?
Yes.
If there's one thing this pandemic has taught us, it's the importance of time in drug and vaccine development. With every passing minute, millions of lives are at stake. But how to speed up vaccine development? By the end of this presentation, you will have answer to this question. We identified a major challenge in this industry. Phase 1 fails to screen out molecules that will be screened in phase 2. There was a huge disparity between the success rates of drug passing through phase 1 and phase 2. What this meant is that all the resources and time spent on 55% of trials when placed. Also, healthy human volunteers who were part of this trial were at serious risk to adverse side effects. We found clinical evidence-based research that additional biomarkers can help us in this regard. This led to the formation of VACO's need statement. Introduce additional screening methods in phase 1 of clinical trials so that companies increase their chances of compound proceeding to approval. Here's the scientific principle behind this idea. We take healthy adult stem cells from the source. These pluripotent cells are then differentiated to derive specialized cells such as hepatocytes, cardiac cells, etc., on which drug testing is done. The cells are under continuous monitoring and screened for morphological and physiological changes for the given criteria of the study. We will be able to test drug toxicity, the right dosage, and also the clinical safety of the molecule, all in a test tube. In conclusion, we decrease testing on healthy humans and use stem cell technology instead to conduct trials in phase 1, thus improve the efficiency of screening process. Our technology will act as an additional filter and lessen the time for phase 2. We got our idea validated from vaccine developers, potential clients, and investors. They all gave us a positive feedback. This slide depicts the impact potential of our idea. Using VACO solutions would decrease the probability of success of first phase trials, which at first sounds bad, but don't worry, because this would increase success rate of phase 2, which has the lowest success rate of all clinical stage studies. This would significantly decrease investment losses, lowering the number of potentially doomed studies in phase 1, but on average, say pharmaceutical companies, from 7 million to 80 million euros per study. The business model is as follows. We will sell our technology and expertise for a monthly subscription to drug and vaccine developers like Grindex and other institutes in Baltic, which would be our beachhead market, and then offer a product to large companies such as Pfizer, Johnson & Johnson. Each year, the global pharmaceutical market is growing with an estimated value of about 2 trillion euros in 2026. Our product is focused on phase 1, where the average growth is about 6% per year, and right now is valued at 14 to 66 million euros in our pitch market, the Baltics. In contrast to its competitors, VACO has to offer much more. Our unique selling point, VACO is the only entity which allows for dose tolerance studies based on real human cell lines, and offers multi-centric usage in a single platform. For our next goal, we plan on making VACO AI-enabled, thus devising algorithms to ensure the right compounds make it through the initial phases of clinical trials. Also, we have a market potential to cater the needs of biotech firms who produce organic molecules and need in vitro testing. So here's why you should pick us. VACO would tremendously increase the likelihood that a molecule would make it to the market earlier and cheaper. Our innovative idea is sustainable, market-ready, and unique. We are the answer. With VACO, we are speeding up vaccine development in the right direction. Choose VACO and experience velocity. Our team comprises of 2 future medics, a chemical engineer, and a biochemist with experience in cellular technology. So yeah, we are very much capable of taking this project forward. Thank you.
Awesome. Thank you, Hiba. Thank you, Team VACO. Do we have any questions?
Hi, it's Anna. Thanks for the impressive presentation. I wanted to ask about the specific cell types, and you mentioned using of IPS. So basically, to test the vaccines, the main cells to test that included in the tests would be T and B cells. So could you comment on the generation of cell types and the other competitors like Prelis Biologics speeded up the COVID antibody production and also vaccine development? What would be the competitive advantage against such technology?
So yeah, the first question would be taken up by my teammate Anastasia, and like the second part of the question, which was on competitors. Yeah, I'll just answer that. Yeah. So like when compared to the antibody production testing, so like the antibodies, like they don't have a long-lasting, they don't provide a long-lasting immunity to the host. In contrast to that, our product would help in like the initial testing phases so that the right vaccine would be developed. I hope that answered the second part. And Anastasia, could you answer the first part?
Can you please repeat first part of the question about T and B cells? Yeah, that was the question. Which cell lines we will use?
Anna, if you can hear us, could you please repeat the question?
Yes, could you comment more on the specific cell lines, because you mentioned differentiating and using the IPS cells. So could you comment more on the technology and the specific cell phenotypes that you would include in your solution?
Yeah, that depends on the drugs we will be testing or vaccines. For example, for a specific drug, if it's connected, for example, gastrointestinal system, we will use, for example, the cells of gaster. And if we are testing, for example, drugs that's connected with nervous system, we will test neurosys. So basically that depends on the drug, and we will differentiate the stem cells to specific cell types depending on the drug. Yeah, for more precise information and more precise result.
Thank you.
Thank you.
All right, thank you so much, Team VACO. Thank you, Hiba and Anastasia. We now move on to Team XR.
Good evening, dear fellows. And today I will address a problem that half of you will be dealing with later in your lives. Hopefully not. By our calculations, we can actually help 90,000 people alone in Latvia. The world's biggest killer is ischemic heart disease, responsible for astonishing 16% of the world's total deaths. This disease is due to atherosclerosis, which has a highly effective drug therapies and precisely known disease pathogenesis. Medications that are the most commonly used to treat it are statins. They are lowering serum lipids to reduce primary and secondary risk for cardiovascular disease. Despite the compelling evidence supporting cardiovascular medications in the prevention of ischemic heart disease, 5 out of 10 patients discontinue treatment. In a population of 10,000 people being treated, 2,000 discontinue treatment because of statin-associated muscle symptoms, which include muscle pain, weakness, fatigue, rhabdomyolysis. Frequently, cause of statin non-adherence and drug discontinuation has been associated with a high rate of cardiovascular events and death. For some patients, 1 out of 10,000, the effects are especially life-threatening. These side effects are leading to low compliance and finally complete stop of the therapy, resulting in lost time, lost money, and lost trust. There are also a high-risk group as Caucasian race, co-disease as second-type diabetes, and most importantly, gender. Women are at a higher risk of statin non-compliance than men are, and are 15% more likely to stop or switch their statin therapy. Therefore, by American College of Cardiology guidelines, they are given statins less often than men, namely losing opportunity of using effective medication. We identified that patients carrying the SLC01B1 variant allele have enhanced risk of adverse effects. This gene is mainly transporter and eliminator of drug from the blood circulation. 60% who reported adverse side effects have this allele. Calculating from the beginning, from all those 10,000, 1,200 stopped due to this allele. These are 12% in total. Our team came up with a solution that can be easily implemented. The blood test assigned by the cardiologist, that's a pharmacogenetic testing, and correct dose or alternative therapy can be chosen, prescribed instead, which shows promise of precision medicine in a nice way. Existing infrastructure can be easily used as PCR or NGS, and the costs, for example, for PCR do not exceed 10 euros, as with other tests. Our next steps will be contacting social security providers, companies, and cardiologists. We have already talked to some cardiologists and heart disease specialists, and they acknowledge this is an existing problem in Latvia, although adverse effects are written off idiopathically. Future development is promising, as around 1/3 of all pharmacogenetic mechanisms are approved in cardiology, and growing research base is revealing more and more genes related to statin alternatives that are used after the. For example, as fibrates, they are used by statin non-tolerated people for patients with diabetes. One more time, time, money, and trust. Also, a new good way for precision medicine. Thank you for your attention, and here are all the team members, 2 medical students, and 1 pharmacological student of pharmacological studies, and all we are researchers at different institutes in Latvia. And questions? Yeah, we'll be glad to answer.
Thank you so much, Anton. Thank you, Team XR. Any questions from the jury?
I have a question. Anton, thank you for your presentation. Could you elaborate if there is currently any alternative for patients who have this mutation, and so they are not the most promising for using statins? Is there currently alternative?
Yes, of course, there are alternatives. So that we chose this field because there are around 10 different medications that are with different pharmacodynamical mechanisms and do not interfere much from one to one. So as I also described, the therapy can be future therapy can be tailored by those.
Okay, thank you.
Anton, one question. Why are you not comparing statins with mildronate? What do you expect from this?
Sorry, I don't understand the relation. I understand the relation, but it's not our topic. Can you rephrase question? In what way do you mean that?
You mentioned a lot of problems with statins.
Yes.
Why do you not try to test mildronate, which have no such problems as statins?
We can try to test them. Yeah, hi, I'm also from this team. And I think it would be a great idea to maybe combine the effects, because but statins are good for lowering the lipid profile, but mildronate would help the oxygen consumption of the tissues. And I think it would be good to test the combination, of course.
Thank you.
Thank you so much, Team XR. Before we move on to the next team, by the risk of sounding pedantic, I would like to remind the jury that it will be easier to put down the marks for the teams as they present, unless, of course, you are taking separate notes. Otherwise, you will have to recall all the presentations afterwards. So, yes, this is something from the organizing team. All right, let's move to the next pitch, and it's Team 5, Tara. I think the floor is yours, right?
Okay, thank you for your patience while we get our slides loaded. Thank you for your time. We're very excited today to present to you algae factors, smoother skin and anti-aging through green biotechnology. So our problem is that consumers who prefer natural products, as per their definition, need an effective and more affordable way to regenerate the skin and reduce signs of aging. Current products certainly don't do that, such as creams, and there are two products that have been proven to do such a thing. One is retinols, which can be harsh and cause reactions for the majority of consumers. And number two is current EGFs, or growth factors. This is a protein naturally occurring in the body that stimulates cell growth and differentiation. However, first-generation products are very expensive to make, 200 to 400 euros per small bottle. They were also produced with the stem cells from foreskin, which created a lot of negative public opinion over the past 6 years. We plan to produce EGF through means of algae using precision fermentation. Precision fermentation is not new. However, a few recent articles dated in 2020 has proven algae to be a proper platform for producing these growth factors. In addition, algae is a very popular ingredient for natural consumers. So the first growth factors were released in 2001. These were a gold standard of anti-aging skincare until about 2014. However, they faced controversy for the reasons mentioned above, and prices doubled in around 2017. However, enter BioEffect. In 2009, they produced a product using barley, and they have experienced about 25% increase in revenue every year with virtually little advertising. So the timing is perfect for us to produce a similar product to appeal to natural consumers using algae. Our MVP will be a 30 and 50 ml bottle in a traditional day serum. We propose that we can take the prices from $165 down to around $70 to $90 through the production of algae. Following that, we plan to produce skincares and lotions. The time is now. The alternative protein market is booming. In fact, recombinant collagen also grew at about 25% demand. Natural cosmetics is also booming, and of course, that was pre-pandemic, but during the pandemic, online sales began to boom. And that is 80% of our business, online sales B2C. The EU is our third largest market. We can start here, then move into North America, where I have contacts, and Asia Pacific, where Priya has contacts. We have two target markets. One is natural consumers. They have an interest in algae already. They have an interest in alternative proteins. Our product can potentially be zero waste. We can use the leftover biomass for biofuel. Also high-end consumers, people who have been priced out of their favorite EGF products and who are allergic to retinols. Here's our business model. 80% will be B2C online sales. We have the personnel. Priya and myself both have experience in website and marketing. 20% will be a white label B2B product. We're in talks with universities both in Latvia and also Berlin, and have some different talks also for R&D that work with algae, particularly in Spain. We have researched several accelerators, International, Green, and Silicon Valley, that would be perfect for our purposes, and we've started making those proposals. Direct competitors, again, traditional EGFs are far too expensive. We hypothesize we could take 60% of the market, and the other products are too harsh or simply don't work. We are currently in the R&D stages. We will be in contacts with labs very shortly to get equipment and personnel involved. Stage 2 is an MVP serum and a lotion. We get a patent registered and applied to our second round fundings that was evidenced in the IP slide. Stage 3 will be marketing and sales. And stage 4, slowly increase our portfolio from serums to lotions and create our B2B product for other larger cosmetic companies. Our product already has proven efficacy, both in wound healing and in cosmetics. We offer it at a lower price point using algae, which appeals to natural consumers. Retinols currently take 10 weeks to work. Our product works much faster, and we can capitalize on the favorable public opinion of BioEffect. Here's our team. My name is Tara. I'm a UX researcher. This is Priya. She is a fifth-year medical student. She can also be involved in the product testing. We plan next to go to Cosmetics Business Live Forum next week to continue to pitch our product. It would be awesome to have your funding and support to bring us into that next stage of pitching. Thank you very much for your time. We would love if you would connect with us on LinkedIn.
Thank you so much, Sarah and Team Biohacked. Do we have questions from the jury?
Yes. Thank you for the presentation. I have a question. I have a couple of questions. So first question is recombinant EGF. It's still a quite big molecule. Do you have any data about diffusion through epidermis, through penetration through the skin, and at which layer of the skin this ingredient would work?
And Priya, are you ready to take questions? You're still muted.
Yeah.
Okay.
Yes, I'll take it. Yes. We are not. I think it is about the epidermal subepidermal layer, and because it has already been suggested to the wound healing, surgical wound healing, and other scarring clearoid factors as well. So I think it is, and also we have data that it is proven there somewhere in the links. Actually, we have linked it there in the presentation. I think in slide 3, slide 2, I guess. Yeah, slide 2. There will be like down in the link we have attached all the data regarding this.
Okay. Yeah. you have a very ambitious project. That's just a small question. And when you're pitching at cosmetics events, you think about this, because a lot of molecules, even vitamin C, there is a problem to penetrate in the skin. And wound healing is different, because there you have a wounded skin, but to apply on healthy skin, it's a lot of problems with penetration. You have to prove that.
Yeah.
The other thing is EGF is actually quite widely manufactured using bacteria, Escherichia coli. That's a very well-established process, and it's actually cost effective. So could you explain more the cost benefits of using algae?
Yeah. Well, in order to release the EGF from the inclusion bodies in bacteria, we propose that that's actually going to be a higher price point, and different accelerators that the 2 articles that we researched to use algae, there's different accelerators involved that would produce that at higher yield at an industrial standard. So we just think that the way that algae grows itself and the accelerators illustrated in those articles, we hypothesize it would produce larger yields, and we wouldn't have to do that extra step to release the EGF from the inclusion bodies with the use of bacteria. Because BioEffect was able to take the price from 200 to 400 to 165 already without that step, since they're not using bacteria. So we propose if we do that with algae, we can produce more.
Thank you.
Yeah.
Thank you, Anna. Any more questions to Team 5 Biohacked?
There's also a link to that article about the inclusion bodies in our.
Yeah, I'm just maybe well extending the question on really advantages of algae, because of course the growth rate is slower than for bacteria, and the question is, have you thought about how to really enhance performance in algae, so to make them more competitive? Because at the moment they are, I mean, depends on the product, but they are still kind of not really the best choice due to the price.
Yes. The thing is, we read on a research paper, there is a algae called Chlamydomonas reharditi, and it is new. Actually, the paper is from 2020. It is, they said that it is favorable and it is good compared to rest, because this growth factors, it is expressed from the chloroplast genome of the algae, and it was written that it is naturally producing some growth factors which is bioidentical to the human growth factors, and it is more favorable compared to the rest. And we have to do the research actually. When we started it from that research paper, and then started collecting information. And also regarding the question before, yeah, I just remembered this human growth factors, it is smaller than the hyaluronic acid, which is the most common compound which is used in the cosmetics which we find in drugstores today. So it is comparatively better than that which is available in the market, the one we are proposing.
And certain hyaluronic acid have actually been able to use nanotechnology to penetrate the skin. So we could look into that as well. And algae has other cosmetic properties that antioxidant, for example, skin smoothing, UV protection. We might be able to present some of those options as well as an additional selling point to using algae.
Thank you.
Yeah.
Thank you so much, Team Biohacked. We now move on to Team 7, Grape.
Hello, everyone. We are Team Peer. Now it is Paula, Evelyn, Alex, and Trenis, and our idea is seaweed bioremedy. Seaweed is a sustainable source of nutrients in diets of human, aquatic, and farm animals. Seaweed provides alternative source of protein for growing world population. It restores aquatic ecosystem and eliminates methane from animals' bodily emissions. The most striking problem that tackles invigorating growth of seaweed is heavy metal exposure from human activities. Our aim is to significantly decrease health risks associated with chronic heavy metal exposure and drive seaweed markets, which the main restraining factor is high cost of cultivation due to mixing with toxic waste chemicals. Seaweed aquaculture stakeholders are producers, processors, traders, and consumers. Seaweed bioremedy is supported by regulation of European Commission on heavy metals thresholds in seaweed. Heavy metal exposure is the main concern of European seaweed producers, who can be considered as potential paying customers. Seaweed bioremedy can be impeded by public misleading opinion that there is no health risks due to lack of short-term symptoms and low consumption of individuals. Our solution is transgenic symbionts. It is favorable bacteria at the surface of seaweeds with genetically increased intake of heavy metals that results in heavy metal-free seaweeds and increased heavy metal detoxification that brings also toxic heavy metal-free ecosystem. Naturally, seaweeds and bacteria are growing in mutually beneficial relationship that is very diverse. For instance, bacteria gets nutrients from seaweed surface when seaweed gets a defense against toxic chemicals. And we would like to enhance this defense specifically to heavy metals. As the most relevant macroalgae, we consider most raw edible seaweeds with non-heavy metal exposure, as Wakame, Kombu, and Hijiki. As the most relevant symbionts, we consider most abundant macroalgae symbionts with engineered heavy metal bioremediation, as cyanobacteria. But why do seaweed bioremedy worth developing if there is ours alternative proteins? Being a natural source of vital nutrients, seaweed also restores aquatic ecosystem and eliminates methane from animals' bodily emissions. And the only main drawback is heavy metal exposure that we would like to solve. Me, Alexei, science and technology, bioengineering and robotics students, Evelina, junior researcher in macrobiology laboratory, and Paula and trainees, students of biotechnology and bioengineering. Thank you for attention.
Thank you so much. Are there any questions from the jury?
Yes, thanks for the presentation. I have a question. Currently for food and feed needs, cultivation of algae in bioreactors in a highly controlled environment where the risks of heavy metals are excluded is widely used. So have you analyzed the price competitiveness and applications of the technology to compare this symbiosis strategy against standardized cultivation of algae in clean bioreactor?
We have found that the most popular way to cultivate seaweed aquaculture is in open sea near the coast, and in bioreactors it's only very small parts, like 1 to 5%, and it's mostly used for cosmetics and not for food, because it's really make the cost really high. And as the second part was about bacteria and symbiosis, so we have found that right now there is some draft experiments how to make bioremediation of heavy metals by cyanobacteria, but the main drawbacks that they like tackle is not to make it in bioreactors, but naturally, and they haven't reached the level of symbiosis. So it is in our area that we can solve.
Okay, thank you. And another like a more like a regulatory aspect. So introducing a symbiont that's in some cases might not be naturally occurring in the same environment. Would it cause any problems, might cause in the ecosystem as it is? And are there any like regulatory restrictions to introduce additional organisms in an existing ecosystem?
Yes, we have found GMO legislations where it's like a framework of European Union, and specifically all of these 5 parts are related to ours. So we need really to take time and understand it more, but based on current solutions, it is possible. And if we take like biochemically how these symbionts like change environment, then I can smoothly talk about how heavy metal resistant works. So basically, heavy metals are taken inside by several kinds of chemicals on the surface, and this principle is almost the same. Is it macroalgae like seaweed, or is it microalgae as cyanobacteria? And on the surface of macroalgae, there is surface of microbes, natural biofilm, and cyanobacteria is part of it. And we would like and cyanobacteria also already genetically modified to intake heavy metals. So we just make sure that our cyanobacteria will be more like let's say focused on heavy metals, not right now we are also sporadically, so we will make sure that they are more abundant. And if you take how this will change ecosystem, so maybe biofilm will be changing and some microbes will be not so worldwide, not so abundant as it is by now. We have found out that it's usually depends on level of toxic heavy metal, toxic chemicals. So if there is more, let's say, heavy metals, and our cyanobacteria are more professional, let's say, in take them and provides this defense to algae, it will be naturally more abundant, and our bacteria will find out in our niche. So it will be like natural selection.
All right, thank you.
So
thank you, Team Grape. And now we move on to the next team. It's Team 8, Proteosmiths.
Hi. Yes.
Hello, Ram. Floor is yours.
Okay. So, one moment. All right. Have you ever felt like you've lost control over your appetite? A feeling of constant craving for sweets or other comfort food? Well, if you have, it is probable that you were or still are amino acid deficient. Want to know how? Stay tuned till the end for the answer. What if I say that 9 out of 10 people in this world are deficient in amino acids? That's a pretty significant number, isn't it? Amino acid deficiency is a frequently overlooked and multifaceted problem. We believe we have a solution. Before jumping into our solution, here are some of the factors causing amino acid deficiency. Insufficient intake of protein is the most common problem. Can also result from poor digestion of protein, which usually comes from lack of hydrochloric acid in the stomach, or could be inherited in the form of biochemical anomalies that limit the body's ability to break down the protein. Last but not least, overcooking. Studies suggest that when food is cooked above 118 degrees Fahrenheit, the high temperature denatures the structure of protein, leading to destruction of some essential amino acids. So the next natural question would be, why does this problem need to be solved? And aren't there any current solutions at all? We try to cherry pick two of the most important problems caused by deficiency of amino acids, besides lowered rate of protein synthesis. And you can see them listed on the slide here. Firstly, lack of certain amino acids can suppress immune responses. For example, the amino acid arginine is an essential part of the peptide chain in the receptor of T lymphocytes. So low levels of arginine can impair the function of T lymphocytes, which are very important for producing antibodies. Also, some amino acids are neurotransmitters themselves, and the deficiency means impaired nerve impulse transmission. The list goes on. The current amino acid supplements in the market are unsustainably produced by breaking down proteins from natural sources or using chemical synthesis producing harmful byproducts. Plus, they're expensive. Here's our big idea. We introduced to you alpha-L amino acids, sustainably sourced, cost-effective, personalized amino acid tablets made using fermentation technology. Our process is fairly straightforward. We first begin by culturing the microbe in its pure form by supplying the preferred carbon source for growth, followed by metabolically engineering them to enable usage of a broader range of substrates and hence higher amino acid yields. The microbes we are especially interested in are Corynebacterium glutamicum and E. coli, especially because their metabolism is well documented and well known. We then introduce the engineered microbe into the bioreactor with optimum growth conditions to start producing the amino acid, followed by purification and drying. For easy consumability, we plan to powder and press the amino acid crystals into tablets. Our major competitor will be Life-Free Organization's Platinum Plus amino acids. Since their production is GMO-free and sourced from vegetables, they happen to be expensive. This bottle of 60 capsules costs $52. Also, they contain a blend of 12 amino acids, which may not be required by all the customers. These are the two main areas where we differentiate and stand out from our competitor. Our product will be considerably cheaper as we employ a micro-based production line, and we also tailor our product to the needs of the customer. Market projections seem optimistic as it is evaluated to be an almost $9 billion industry by 2026 with a compound annual growth rate of 7%. Nutraceuticals, dietary supplements, and convenience food are the main areas where we observe demand. And finally, here's a business model. We plan on establishing collaboration with laboratories for testing our customers for amino acid deficiencies and with industries requiring amino acids to fortify their products. This will be done in parallel with setting up a small-scale manufacturing plant. Once we produce our initial product, we essentially use it as a bait to attract investors to enter the market and drive the supply chain. We are the Producemates, a group of ambitious students with a vision to create a healthy and active lifestyle. So what are you waiting for? Join us and let's help make this world a better place. Now back to our initial question. So the answer is that the deficiency of certain amino acids, which act as neurotransmitters, would mean that the feeling of fullness cannot be conveyed to the brain and hence the craving. This marks the end of our presentation. Thank you, and we'd be glad to answer your questions.
Thank you. We have questions.
May I ask, thank you for presentation and good idea. can you elaborate a little bit more on your business scale-up strategy? How you see that?
Yeah, well, so essentially we plan on setting up a small-scale manufacturing plant, and when we have our initial product and initial results, we essentially prove that we can produce the product at a lower cost, and we attract investors to scale up our production. That's our preliminary plan.
Yeah, thank you.
Hi, I would like to also ask a question. Could you maybe elaborate a little bit more on what kind of regulatory barriers would you anticipate to face before launching your product?
I would expect some kind of barriers for genetic modification. I have no current expertise in this area. Can you guys back me up on this?
The research doesn't need, there is not a lot of barriers in the research. The barrier starts when you going in market and when the customers can access these things. But for supplements, it's not so hard. It's like most of 2 years of paperwork, at least in European Union.
Thank you, Robert. Yeah, I hope we managed to answer your question.
Thank you.
Thank you.
All right, thanks, Team Proteosmiths.
Thank you.
Now we move on to Team 10, Clean Team. Are you here, guys?
Yes, hello. We are the Clean Team. I'm sharing my screen. And, okay, so our team is operating mostly in the field of business and biotechnology. We are Yila, Kaiwa, Edmunds, and Lev. And we were choosing to deal with the environmental health issues. So we were thinking about what would be the most effective or the most important to deal with. So we ended up specifically dealing with soil pH, because soil pH is one of the most important properties of the soil that is affecting all the biological, chemical, and physical reactions. So without good soil pH, which should be around 6 to 7, we don't have healthy crop growth. So, and also we don't have a healthy soil, so it affects our lives as well. So how it's addressed, usually they are using limestone on the field to improve soil pH, because most of the soil is acidic. But the problem is with this that the limestone supplies are depleting. For example, here in Estonia, where I am, it looks like that in few years there will be just not enough limestones anymore. And by 2050 in Europe, that might be a huge problem. So also the quarries and how limestone is at the moment, how people are getting limestone, it is not energy efficient, and it requires a lot of disruption of land, and it's just not sustainable. So our solution was focusing that how we could produce sustainably limestone that would affect the soil pH and soil health. So what we came up with that we could use for this construction waste, and we could use on the construction waste bacteria with the required nutrients. So we could use it through integrated carbon mineralization and create agricultural limestone for that, what could be used for enhanced rock weathering. There is already a solution for limestone producing bacteria, but they are using it in building constructions. So it is used at the moment as a self-healing concrete. And we would use or that what is already existing and work on that existing bacterian solution, or we could work on our own solution. So how it would look like in practice, we would get the construction waste for the companies. They are already paying for the construction waste recycling, and we would add the bacteria which is feeding on that construction waste and producing limestone. And in Latvia at the moment there is 2 million hectares of total farmlands. From that 1,700,000 is conventional farms. And in Europe, there is a goal to reach by 2030, 25% of the agricultural lands to become organic. And the sustainably produced limestone could help in that, because when they are using it through enhanced rock weathering, then it is also capturing CO2 from the atmosphere, a huge amounts of CO2. And that can be taken into the lands, and it is helping soil microbiomes and plant roots to grow better. So there would be also less need for use of fertilizers and additional things like which is just not that much needed. And so the benefits would be to create a sustainable limestone supply, to recycle construction waste, and also reduce carbon dioxide from the atmosphere, and through that achieve the optimal soil pH for healthy plant growth, microbial diversity protection also, and restore soil organic matter, because through the optimal pH and added CO2, the soil microbiomes are also working faster, and the decomposition is happening faster. So it would be really healthy. And the next steps would be to work on this growing bacteria, test it in laboratory, and then test it on the field. So for that, we could develop partnerships with universities and laboratories to work on this solution. Here are our references, and thank you for the attention.
Thank you so much, Clean Team. It's time for the questions.
May I ask, do you have any proof of concept somewhere demonstrated at reasonable scale that this would work?
Yeah, at the moment there is that limestone producing bacteria. They are using it only in the buildings. So when there is some crack in the buildings, for example, then that bacteria is feeding on the concrete, producing limestone, and this way it's healing the cracks. And then, yeah, this is how the idea came, that we could actually use it in this way.
Yeah, I'm asking about the scale, because what is done is, I mean, talking about relatively, small type of cracks. You are talking about, I think, this kind of converting materials from, talking a big mass, what you need to process. So I wonder if how sure you are this might work at this scale. I mean,
yeah, we should definitely test it like first in a smaller scale and then on the bigger scale. So at the moment, yeah, we need to work on it in the future.
And maybe second one, do you know, is there any limiting factors for these bacterial growth? As far as I remember, it was that you need urea or some kind of substances for this. And
yeah, it can be
limiting and expensive.
Yeah, it can be lactic acid, or I think Kaiva, my teammate, could help in that what kind of nutrient it might be.
These limiting factors, as these bacteria are extremophiles, which are prone and can survive these harsh conditions, such as concrete in the concrete healing bacteria. The only thing they really need is some kind of nutrition so they can stay alive and then produce this limestone as a byproduct by catalyzing the fixation of CO2 to the calcium in the construction waste. But yeah, one is this calcium lactate. That's one option as a nutrition source, but there can also be other opportunities that we can yet to explore. And I would like to mention about the scalability. Yes, this is very theoretical still, so we would have to actually test it out on the field to see, let's say, how many bacteria is needed per cubic ton of construction waste to produce the wanted product. So I'm afraid we can't really answer this question at this moment of time. Yes, I hope this answers something.
Thank you. Thank you.
Could you tell me about pH changes during the day, week, or month? Because production of limestone is connected with pH changes.
Yes, they are using limestone in mostly acidic fields. So, and mostly that is the problem with the soils, that most of the time it's more acidic. So limestone needs to be added, and they are using it for example, in Latvia, that would be used like 1,007,000 tons would be needed per year. Did I understand the question right?
The question was how bacteria work. How are the changes of pH values before they start and after 1 day, week, or month?
Yeah, about the speed of the reaction of the whole soil becoming more alkaline with the added limestone. I'm not entirely sure, but it's definitely not a day's work. It has weeks of, I guess, these biochemical processes to be integrated, and then the whole soil becomes more alkaline over time. Yes.
Kurš no visiem? Jā, viss kārtībā, jā.
All right. Thank you so much, Clean Team. Now we move on to the next team, 12. Welcome, Wasted Gold.
Hello. Could the person with an open mic turn it off, please?
Yeah, all right. Sorry, we've muted everyone. Ugis, you're good to go.
Thanks. Okay, we're team number 12 and we're named Wasted Gold. Our team is made up of 2 biologists and 2 chemists. The problem we thought of is cobalt. Cobalt is primarily used in lithium-ion batteries, magnetic wear-resistant steel alloys, and multiple types of catalysts. And as you can see, cobalt demand is expected to skyrocket. At today's price, it's about $35,000 US per ton. And it's practically grown by 50% over the last 2 years. And as you can see, there's a definite need for it, and it's one of the most needed resources today. But the problem is, how do we acquire cobalt? It's mainly done through manual labor and just in mines, which cause a lot of disruptions in local ecosystems. They degrade quite rapidly and cause flora and fauna die-offs. Cobalt poisoning causes severe developmental disorders in humans, so any exposure to cobalt salts is really unhealthy. And another problem is that depleted mines are not properly decommissioned. Once it's become unprofitable, the land is just left as is. There is no decontamination processes in play, and there are pretty much no regulations about this. And another problem is, if there is something left to contaminate the waters downstream, there's also materials to remove, to extract and get the money for. Therefore, it's needed to clean up these messes. And our solution is using plants. Basically, there are a few types of plants which hyperaccumulate heavy metals. We've selected these, seen papers describing these two plant species, which are cobalt hyperaccumulators. And basically, once you get the biomass in, you clean up the environment and have some cobalt to extract. Once you have collected your biomass, you just have to burn it. And once you burnt the biomass, pretty much the ashes are high quality ore. And honestly, they're better ore quality than what's considered high quality today. And once you've got this ore, you obviously can use whatever means you want to purify it, either through electrolysis or dissolving it and recrystallizing it as salts or other methods. And once we use this method to decontaminate old mining sites, we basically increase cobalt yields, we decrease impact on local ecosystems, we rehabilitate them, and these mining sites can be used for other tasks. Also, there's less toxins in the environment, therefore local communities of animals and people benefit, and there's complete exploitation of these mining sites. So there's nothing left behind. And of course, it's sustainable. And people who might want these are obviously ore producers. Why wouldn't they want to increase their yields? As well as environmental activists who want to have a cleaner, safer environment in their own little ecosystems, as well as countries housing these cobalt deposits, and of course cobalt-dependent industries which rely on cobalt production. And if you produce more, there's bound to be some kind of a price decrease. And the key takeaways are that 6 countries hold about 85% of the world's cobalt production, but Congo is the greatest of them all, and they hold roughly 50%. And mining requires a lot of human labor, and it's done mostly by untrained professional people. So there's a lot of exposure, and it's done manually. And of course, the depleted mines still contain a lot of resources which we can exploit. And there are practically no environmental protections, as we've seen thus far, about these decommissioned sites. And of course, our solution is great because we get 2 birds with 1 stone. We produce resources while cleaning up the environment. And well, we just want to make a simple, we want to produce cobalt in a greener way. Thank you for your attention.
Thank you so much, Wasted Gold. Are there any questions from the jury?
Yes, thanks for the presentation. I have a question. So each different plant species requires different growth conditions, including soil quality, climate conditions. For those specific plant species that you mentioned, what is the climate and what is the growth area where they grow? What is the growth area, and is that from the region where there are actually the cobalt mines, or those plants need to be introduced there?
Some grow in regions where there are cobalt mines, yes, in Australia, and some might be introduced in Africa. They mostly grow in arid climates and subtropicals.
Okay, thank you. And another short question, do you know what is the yield, like how many milligrams per biomass of the plant you can get actually the...
Yes, there was the data here.
Okay. Okay, thanks.
Any other questions?
Right, I think we're good. Thank you so much, Judith and team Wasted Gold. We are now moving to team 13, Marcella. The stage is yours.
Hi. Hello, everyone. I'm really happy to present today our team's findings during the hackathon. So, to innovate, we must collaborate, and data is the driving force behind the innovation. So we must remove challenges for data access and usability to accelerate collaboration. My name is Edgar, and I'm presenting Team Marcella to show you a machine learning solution that allows to create value from human biological and omics data. So we are acquiring lots of human biological information about the genetics to understand how human organisms work. But because this data is very related to the person who provided it, there are privacy and ethical restrictions to use it. But because of this, we can't share this data and allow access to others to create value and solutions to make our human lives better. So who are those who would benefit from solution that tackles these challenges? At one hand, there are biobanks and medical institutions who gather and store this information, and they are unable to provide it further. On the other hand, there is biotech research and pharma industries who need this information to develop products, solutions, drugs, and make their businesses grow. The estimate for market size for data sets and AI training was evaluated about 1 billion in 2019 and expected to grow 4 times till 2027. So what is the solution? We could create synthetic data that is computer generated and preserves the same relations as original. But because of that, it doesn't relate to original humans. Just like the pictures with faces, genetic data represent something who we are. And synthetic data that represents the same thing, but because it's not real humans, there is now this problem. And with computer methods, we can learn what are the attentions, what are the specific features that makes this data set so valuable. So, and because of synthetic data, it's more privacy concerned because it doesn't link to original subjects. It hasn't lost its information because of anonymization or pseudonymization. Then it's more usable, just like the original data. And because it's machine created, it's scalable. you can use it to train models or make more data with examples to make better products. So our solution would be a software platform that has a synthetic data generator, and then add different modules that add value to these created data for AI training, data analytics, data sharing, product development, and testing. Our business model would be at start to work with industry partners to create custom solutions for them and concentrate of specific data sets, for example, genetic and their phenotype relations that has been shown have correlations in these data sets. Then we would create software platform that is easily usable for data scientists and researchers with user interface, and they could pay and acquire it. And then the last part of our idea is an online platform where we add our cloud computing capabilities, so people can try these data generators and see how can they use them. So our steps after hackathon would be collaborate with science and validate our solution, working on research, then add modules for synthetic data analysis, create user-friendly software platform that specialists can use without programming knowledge, and add our computing capabilities. So our team has competences to do this. We have competence in machine learning, bioinformatics, IT software regulation, law, microbiology, and research and industry connections. So to recap, genomic data is sensitive and it's hard to share. So our solution is synthetic data generation that provides analysis models and creates value for industry and parties. Thank you for listening. I'm ready to answer to your questions.
Edgar.
Yes.
Thank you very much for the presentation. So the field of synthetic data took off a lot around, for example, self-driving cars, when it makes sense why generating synthetic field data will help you to train a car. Do you have an example of a use case when you can leverage synthetic biological data in order to solve a specific solution?
For example, you want to see if some kind of specific group has some kind of characteristics for human phenotypes. There was a research that showed in Estonia that about link between human eye color and original data and synthetic data. So, for example, if you want to study some kind of illness, you can get access to the information. What are these variations in human genetics that are influencing this without compromising the privacy of humans who are provided this information? We could find new places how to develop solutions, or maybe train, or maybe biobanks want to expand their capabilities and they need to hire someone to create a solution. They can't give the original data, so they create synthetic one.
I can maybe also chime in a bit, building on what Edgar just said. So one of the during the hackathon, we held a conversation with the senior researchers from Biomedical Research Center, and one of the use cases that was highlighted over the course of these conversations was that the biobanks essentially hold a ton of data. And there are companies who would like to engage with the biobanks and build their product solutions based on these data. However, it requires a ton of regulatory adherence and documentation and ethics board compliance, etc. So for these companies, before going through that documentation or administrative hustle, it would make their lives way easier to play and pilot or do proof of concepts, first of all, on the synthetic data sets. And then if the first validation has been done, then they might proceed with actually engaging and going through the documentation, compliance from ethics board, et cetera, et cetera. So this would essentially make the life easier for biobanks collaborating with pharma companies, or pharma companies collaborating with biobanks and other data set holders.
Thank you.
May I ask, what are the competitors in the market, and what makes you so unique, and how do you try, if there are competitors, how do you going to keep them at bay?
The competition is very little, and it's more used in financial sectors, in banking, where they have private client data, and they want company to come in and create solution, and they create synthetic data for them. The lack of competition is because this is very specialized field, and not really, and there is people haven't get grasp of what we can do with the synthetic data, and they want to specialize these algorithms for specific research purposes and collaboration. That hasn't been tapped into it yet.
Maybe I can also add a little bit. So what Edgar just said is that finance and insurance, where this technology is applied more, is way lucrative field than biotech currently. That's why most of the companies have gravitated towards that, and there's sort of green field in the biotech space. And that's why we have implemented within our business model, not only providing the engine for synthesizing the data, but also adding a layer of service that would be model creation, data analysis, just to build the relationship and capture these relationships in order to establish and grow the business further. And in this way, we could prevent our competitors in claiming our market shares eventually.
Thank you.
All right, thank you so much, Team Marcello. We now proceed to the final presentation. This is Team 15, Biocult. The floor is yours.
Thank you. I will try sharing my screen now. Hi, everybody. I'm Christopher from Reocto, the cool reactor. When have you ever found yourself in this situation where you don't have enough money? We have as well. And especially in the laboratory, when we are in the process of scaling up a bioprocess. We start from the Falcon tube, we go to the flask, maybe a bigger flask then, but at some point we want to go and get this really nice actual bioreactor. But our wallets don't think the same as we do. And this is where bioreactor comes in. This is where Reocto comes in, sorry. We have to adapt normally to what the university has bought, the bioreactor that we have available. Maybe it's many years old, maybe it's too big or too small for what we actually need, and doesn't have the functionalities that we actually want to. Or maybe we are a startup and we want to make a proof of concept, and we don't want to, dilute half of our shares buying a new bioreactor that does a lot of stuff that we actually don't need. So that's where Reocto gives in value. I want to tell you the story of Brigitta, who was a PhD student, and she needed a bioreactor, which could cost tens of thousands of euros potentially. So they did not have this budget, of course, as most of PhD students, and they went to Depot and bought a series of construction materials, built their own bioreactor. This, of course, took a lot of time, around 2 years, and some money. It was cheap, but it still took some money. But finally, they could make a proof of concept. They got 2 papers out at least, and she graduated a month ago from one of her PhDs at least. And so this worked, but it took a lot of work. They were missing Reocto, the cool reactor. What we offer is an affordable solution. It is absolutely easy to use. It is totally simple, user-friendly, and on top of that, you get free consultancy for life. When you buy a bioreactor, you are buying a lot of stuff that can actually be reduced in cost. So we take out one of the most expensive parts, which is the control module, and we put it up in the cloud. So now all the processing power, it's up to us. You just get your bioprocess done. You can, as a benefit, you can control it and monitor from anywhere, basically phone, notebook, your office. But also we offer modularity, which we think it's our most valuable asset. Our bioreactor is designed in a way that you can get different features, different accessories, and you can build your own bioreactor to fit your own actual needs and not buying extra features. So as you can see, a lot of parts are also made out of plastic. That reduces the cost. And also here you can see 3 different flasks, and different modules are put in or out of the bioreactor depending on the needs. The bioreactor industry is a $5.4 billion market, and we specifically want to address the PhD students and early researchers, and of course startups trying to make a proof of concept. We offer a way more flexible, from the modularity point of view, and way more cost-effective solution than there are already in the market. Before the question and answer, I want to introduce my team. This is Krish, microbiologist, Philips, biotechnologist, and myself, a molecular biologist. I'm open for questions. I know I have run and probably have a lot of time, but.
Christopher, thank you very much for the presentation. Totally can relate to the problem that you presented. Fantastic. And it also sounds like a very good solution. Now, usually when we see something which is a very clear problem and a very good solution, we ask ourselves, why no one has done it before? If it's such a clear solution, such a good idea, why nobody has done it before? Do you have any hypothesis or any thoughts about why no one has done it before?
Yes, that's actually our first question as well. Discussing it in the group is like, this is so cool. Like, why nobody has done it? We strongly believe that this comes from the approach that the companies have. that they are aiming to make a long-standing, long-term product that is probably lifelong product that you can use for many years, and that has to be super precise, super exact. It has to be most of the time, for example, for medical purposes, so it has to meet very high standards. Our approach is for small research or the beginning of the researches, also for startups trying to make a proof of concept. So exactitude and high standards are actually not our aim. So we are getting a lot of high costs out of the equation, just sticking to making it work. So our thing is, you buy this, you get, it only works for a certain amount of time, but you will get your proof of concept done. So you will get your PhD done, or your startup can go and show the investors, here's what we're doing. It's going in the right direction. If you give us money, we will go and buy like Sartorius bioreactor. I hope that answers the question.
Thank you very much.
Well, may I ask, well, still, I mean, you have, for example, Lambda reactors, bioreactors. They are also kind of tailor-made, and they are at the cheap end. So what makes you better, for example, as Lambda?
I will give the microphone to my teammates to also answer that question. I guess Philips can answer.
So in general, our approach would be that we would be cheaper, but actually we exactly at the Lambda's competitor so far we identified Applicon, who shifted a lot during like the last 1-2 years to the single-use bioreactors, like plastic ones as well, but so with consoles. Do you have any like price for Lambda? Because this solution we would hope to be between 1,000 and 2,000 euros.
Okay,
so
I don't have it on top of my head, but it was on the cheap end. Yeah, well, just you know, just because the concept-wise, they have the same idea. You know, they have a very basic kind of container where you stick in all the sensors, what kind of sensors you have, and you have a mixer. So, but you are aware of this, and so what is kind of edge what you are proposing comparing to that?
So yeah, well, once again, for about Lambda specifically, but in general, our cost reduction which costs a lot, sometimes more than, in many cases, more than reactor. We believe that it reduces cost, but I'm not sure what Lambda.
Okay. There's some problem
yeah,
with the connection.
Yeah,
anyway,
it's breaking up, but basically we are taking the most expensive part, which is the control module that sometimes costs way more than the actual mixer and hardware. So all this, let's call it CPU, that we take it out and we take care of everything that we can software-wise to the cloud. So we are centralizing that. Then you're not paying for the software. You're only paying for the actual hardware, which is the cheap part.
So the saving is on the controls. I mean, on the control part. Okay.
Yeah. Yes.
Okay. Okay. Thank you.
Thank you so much, Team Biocult. Thank you, Christopher and Philips. So these were all the teams. Thank you once again for your presentations. Right now, the jury will convene in a separate breakout room. I will just push the button, and the jury will automatically be directed to a different room. And Igors will be there from the Biocatalyst team to moderate the discussion. And meanwhile, we will have a small request for the participants of Biocatalyst. All right. The breakout room is in process. I will share my screen now. Can you see the slides? Good.
Yeah.
Whew, that was really cool, ladies and gentlemen. I'm really excited, really eager to know who are the winners. It's still out. We don't know yet. The jury is deciding. While they're doing that, we have a small request for you, and then a couple of announcements. So before we proceed, we would really appreciate if you could go to biocatalyst.eu/form and answer a couple of questions about basically your opinion about the Innovation Lab. And that would help us greatly improve the next iteration next year. So I will also paste the link. Oh, sorry. I will also paste the link into the chat. If I find the chat. Yeah. So once you do that, please come back to this Zoom call, and we'll go on with a couple of announcements. Thank you.
Andrei, can I make a question about the form?
Sure.
The second question says, please rate the Biocatalyst Innovation Lab program. I guess this just this hackathon, right? This referring to this program that we just went through.
Yes, of course. So the last 5 months.
Yeah,
this
cool.
is it. Yeah. Only the parts that you witnessed, of course. Don't try to rate the parts you were not present on. Thank you for the answers. The jury is still discussing the results. It's a tough choice. So I guess it's just a couple of more minutes and then we'll continue. All right. So a couple of announcements from us, from the Biocatalyst team. There are a couple of things that we've been working on in the last half a year. And the first thing that we wanted to share with you is our emerging membership in the Young European Biotech Network. So we, as the Biocatalyst Foundation, a non-profit entity that's registered in Latvia, are becoming a part of YEBN. And so everyone who is a part of Biocatalyst, that means you, so you are the alumni of the Innovation Lab, can join the YEBN network absolutely free of charge. And what's in it for you? It's, of course, new connections, new job opportunities, seminars, meetups, other events across Europe, and a chance to meet awesome people who have similar passion. Next thing we would like to mention is that Biocatalyst is planning to assemble the first ever iGEM team from Latvia in 2022. iGEM is the largest synth-bio competition in the world. It means internationally genetically engineered machine. And so if any of you is interested in being a part of the team or helping to mentor the team or otherwise help with organizing this is a big thing. So we will need some help as well. Do get in touch with us and we'll discuss how we can collaborate.
When does it start, Andrew?
So this year's competition is already underway. So we are assembling a team for the next year. We will start in the autumn of this year. And so the team should be functional by the end of the winter 2022. So something around February 2022. And finally, another thing that we've been busy with in the recent time is the so-called Biocatalyst Conference, which will be a one-day in-person event in Riga in March 2022, which will be fully dedicated to biotechnology, gathering together around 300 participants from all types of stakeholders, from industry and startups and venture capital to academia and policymakers, and all the alumni of the Innovation Lab 2021. You, ladies and gentlemen, are invited and you are eligible for free tickets, which we will send you more information about a bit later. Another thing to add here is that we are actively looking for people to help us with the organizing issues. And so if you are interested to take part in this direction, please also do get in touch with us. Right, I can see a lot of things in the chat. Let me just. All right, cool. So without further ado, everyone is here. I guess the jury is here. We can move on to the final part of the finals. Igor, it's you.
Thank you. Yeah, jury had quite a difficult task selecting the winners because teams did a really good job, and I have to say you progressed since Wednesday, since we had the first presentations. So well done on putting an effort. And we'll start first with a prize from.
Bet 5002 komandų išsidalino. Du PM. Tipo, kad laimėtojai pasiims 5000, bet iš tikrųjų tai pirmos vietos laimėti 2,5, antros vietos 1,5.
Evelina, could you please mute yourself? Oh, thank you. So we'll start first with a special prize from Malaria. They have a special prize for one team and one team for the winner, but we're not announcing the winner yet. So Anna, could you please announce your special prize who you're giving it to.
Okay. First of all, I would like to thank all teams for the presentations and the work you have done. This is very interesting evening for me, and it's a pleasure to see a lot of new ideas and new solutions that some of them are theoretical, but I really hope that they will be brought to life and we will have them in the following years. For Malaria Cosmetics, we will give a small, like a treat and a prize for the winner, but we wanted to award an additional team that I liked a lot, and I have seen a little bit of the progress during this all the program as well, and I really see the strengths of the team, and even if there will be some pivots and changes in the product and the applications, I really see that this team will be definitely someone that will kick the cosmetics field. So Malaria Cosmetics special small prizes will go to Biohack team. Congratulations, ladies.
Congratulations.
Thank you very much.
And we have one more small prize before we get to the main prizes. This is a prize from a Latvian investment agency. Anna, could I ask you to tell which team would you like to give your small prize to?
Yes, I wanted to say thank you to the organizers and also for the teams. It was a great event, a series of events, and I'm looking further to the developments of Biocatalyst and also biotech community in Latvia and in Baltics. Our special prize goes to Team Marcella, as we see that accessibility of genomics data for innovation purposes is still a huge problem. So we want to give something for this team, and I hope that synthetic data at least somehow will tackle this problem. Thank you.
Thank you. We are very honored to receive this prize. We are happy.
Thank you.
Thank you. And before we jump to the final prizes, top 3 teams, we would like also a small announcement, not announcement, a little prize from a Biocatalyst Foundation. We're starting an incubator. We don't announce yet who is getting there, but we'll contact the team who would like to invite to be part of the incubator. This is for later. We will get in touch with you. And now the final prize. Talis, could you please announce the winners, starting with the place number 3?
Yeah, thank you. I will start with the runners-up, and but before that, I would like also to thank Biocatalyst with the whole team, really helping to promote biotechnologies, bioengineering in the Baltic region. I think this was a really useful start, and I hope this will continue. I think that the prize, which is actually coming from Student Innovation Grant Scheme, is more about promoting student innovation capacities. And I think this is very remarkable that in several of teams we had students from Latvian University, Technical University, other universities engaged. So I think this is a new experience for you. And now I'm coming to the prizes, and the third position with a very first competition, but still is going to the company Biohack. So this is number 3. Congratulations. The next, okay. Then number 2, again, they were really competing point to point, but still the winners, the number 2 position is the company, the team Marcella. That's number 2. Congratulations. Thank you. And then, of course, the final or the most important prize, number 1, the winners of this competition, actually anonymous. They were not really much doubt about this team, because they are proposing quite viable actually idea, and not only idea, but the product which will be, seems to be very close to the market and to be very useful also for students, but not only for them. So the winner is a Biocalt. Congrats.
Well, congratulations all the winning teams. Congratulations to all the teams who took part in the competition. We will get in touch with you about the details, about the prizes, and etc. etc. And well done. Okay, Andrei, I give words to you now.
Whoa! Congratulations, everyone. Really excited to hear the results. Just a couple of less exciting stuff. The boring details. We will contact, as Igor's mentioned, we will contact the winners separately regarding the monetary prizes and the special prizes as well. We'll also send you, all of you, digital diplomas, which you can flaunt on social media, etc. And speaking of which, you are of course also encouraged to add the Biocatalyst Innovation Lab to your LinkedIn profiles if you wish to do so. We'll send you the link to our LinkedIn page as well. And again, as Igor's mentioned, we will also get in touch with some of the teams regarding whether you would like to collaborate with us in the Biocatalyst Incubator. Other than that, we, the Biocatalyst team, would like to say a huge thanks to all the participants for your dedication, perseverance, and creativity, and your desire to learn something new, to meet new people, and to create some new ideas. And of course, we would like to make a big shout out to all our partners, sponsors, and friends. Without your help, this would not have happened. Thank you to everyone. The next iteration of Biocatalyst, which is happening this fall, is definitely going to be bigger and better. So to everyone, have a great summer, and hopefully we'll stay in touch. Cheers.
Thank you.
See you.